And so next we have Dr. Wipawee Jan, who will be leading us into the workshop for you to try out our tool, web-based tool. So if you have your laptop, we would encourage you to bring it out. Otherwise, you can access this using your phone, but it wouldn't give you the best experience.
Good afternoon, everyone. My name is Wipawee Songsaeng, or you can also call me Jan. And today, with these sessions, I want to go through with you the tutorial and the hand-on use of GRC mappers. So as Piranya was saying, if you have a computer, I would like for you to take it out, and we will work through this together. I just want to say that during these sessions, if you have any questions, or if I talk too fast, if I go too fast, and if you need any help, just raise your hand or give us a signal so we can go and assist you at this time. So yeah. First, before we begin, I just want to go to our GRC tool very quickly with you. So when you visit our GenRe-Mekong websites, you can see that we have different tools. The first one is the grcMalaria package. So for this one, you use the code to run and turn the genomic data into the meaningful graphical visualizations, which make it easier to read and interpret. But for today, what we're gonna work on together is we're gonna use the GRC Mapper web app, which is a quick exploration for the public surveillance of the GRC data. So this is very useful for the NMCP activities. So we will go through this together today. So wait. So when you open your browser, I would like for you to type in genremekong.org. So we'll go through this together. Just type in genremekong.org. So I just first briefly go through this website with you. So when you go to our GenRe-Mekong page, you can learn about what we do by looking at our project here. And when you scroll down the page, you can also check what we have in this site. And when you go down even further, you can see the data that we have available, as well as the tools. And we also have like a previous workshop and training that we did before for you all to see. And when you go down to page, you can also see our latest news here, if you're interested to look at our news. And also when you go down, you can see the videos explaining about what we do here in GenRe-Mekong. And at the end of the page, if you are interested, you can look at our feature publications in case you're interested to read our papers. So for today, what we're gonna do is we're gonna go to the top of the page. And when you look at the top right corner, you can see the yellow icon here with the GRC mapper. So we're gonna click on that. Okay, hope you are following. Okay, so it will direct you to this page here. So here you can see that we have the data which are available for exploration of this web application. So this data came from different publications. So we use the global data set of PF7 malaria gen, and also the first paper that we published in the GenRe-Mekong project. You can also see our feature here and the short video explaining how to use the GRC mappers. So in case you can go back and look at the video as well. So now we scroll down to this, and then we're gonna see our first visualizations here of the graph for sample collection from Cambodia, Vietnam, and Laos. So can I ask if we are all in the same page? This one. So is everyone seeing the graph already? Yes? Okay. All right. So before going into more detail, yes, okay. So I'm just gonna quickly explain, spend some time to talk about the user interface and the selection on the left-hand side here. this box. So here on the top, you can see 3 options. We have graph, single map, which will display a single map, and also compare maps here, which will display 2 maps at the same time. So when you look down at this box, you can also see the sample selection and also the feature selections. So we can interact with these letters when we're going through our exercise together. So for example, when you click on the selected samples, on the edit at the selected samples, how we are following, we can see the list of different countries, different states, and also selected studies and selected different period of time. So here, you can make the change for your sample selection. For example, when you click on the countries, you can see the different list of the regions at the top. When you selected the regions, then it will select the whole every country in that regions. But if you want to select one by one, you can scroll down and go through each country and select the country one by one. So for example here, if you click on the country selection, all the remaining field below will also be updated. Okay, let's take some example together. By selected only Cambodia, so you just click deselected Laos and Vietnam. And then if you can see that the province has been changed and all those studies and the period of time, all the fields have been updated. So if you want to see the change, then we will go and click on display changes. And now you can see only the sample collection from Cambodia in different provinces. Okay, so are we all in the same map now?
What is the difference of compare map? Map and compare map, single map and compare map.
Yeah, we'll go through the letters. So compare maps, you're comparing 2 maps at the same time.
Or mean that you select 2 countries?
You can choose, different country or different map. Like if you want to see drug resistance and you want to see sample count, you can also compare different features of the map as well.
No, I'm.
Yeah.
We will go through it. Yeah.
Oh yeah,
We'll go through that later with this. Yeah. So this is the first step. Okay. All right. So that is just the example of the first step for the sample selections here. Okay. Now I would like for all of us to go back. Click edit again at the sample selection and selected Cambodia, Laos, and Vietnam for our analysis. Is it too fast? So yeah, we'll go down to we'll go back to our original map of Cambodia, Vietnam, and Laos. So I was showing you how to select the samples, but now we're gonna go through the feature selections. So what we're gonna do is we're gonna go down there where it says selected features and click on edit. There. Okay. And then when you click on the selected features, you will see these 4 features coming up. So I'm gonna explain to you. The first one is the sample count. So the sample count is what you already see in the graph. Is all the samples including in the graph. And the second one is the drug resistance. For these, it will give the percent of the sample which are predicted to be resistant to the selected drug. And mutation prevalence, we will show you the mutations. And allele proportion will give you which allele associated with which resistant gene. So for example, when you click on drug resistance, you can see here that we will have a list of 10 different drugs which you can select it from in case you want to see a different resistance. Just to note that when you change the selected features, the field will be updated. For this, you can see the drug resistance here. And that will be the same for allele mutation. mutation prevalence. So when you click on it, you will see different mutation here. And when you click on allele proportion, you can also see a different allele here. For the mutation prevalence and allele proportion, you can click on the heading here, and it will show you the tables of which allele or which mutation associated with anti-malarial in case you forget which allele or which mutation is associated with which drugs. So you can also come back and check on that. Okay. Okay, so you're getting this. This is like how when you selected the sample. Okay, so the other features is graph displaying. So you can see in the box that you have a display button here. So when you click on it, you can see the display of the countries of your selections in case you wasn't sure which country you select. Same with the province of your selection and the studies, and also the year and the map or graph features that you have been selected. So you can always come back and check on this box here in case you wasn't sure which selection did you have made. Okay, so the other interesting about this graph is that when you look at the right corner there, you can see that you can interact with the graph. For example, if you want to remove Cambodia from the graph, you can just click on it. And now you can see that Cambodia is removed from the graph. And for example, if you want it back, then you just click on it again and it will come back. Okay, now if you want to see the sample from only Cambodia, what can you do? You can just double click on that and it will only show you the sample from Cambodia. So you're kind of able to play with it a little bit and then see what you want to show on the graph. Okay, so I'm just gonna give you the brief summary of what we just did together. So on the first option on the top, you can select whether you want to see the map or the graph. And in this box, we can make a sample selection and feature selection. So you can kind of adjust this box according to your selections. And on the right side, you can see, you can choose what you want to see on the map by click on the display here. Okay, so we're gonna produce our first graph together. I'll just try to go slowly. Okay, so we will go to our sample selection. So we click on selected sample here. Okay, and then we're still gonna choose the 3 countries, Cambodia, Laos, and Vietnam for our countries. And we wanna reduce the time period, so we're gonna choose from the year 2000 to 2019. So we're gonna change the time period a little bit. Then click on display change. Okay, so now you see the graph of the sample collection from Cambodia, but we want to change from the sample collection to the drug resistant graph. So we're gonna go to the selected features on the bottom here. And yeah, we're just gonna choose drug resistant. Okay, for this time, we're gonna choose artemisinin resistance. And then click on display change. Okay, so now you can see the graph of the percentage for the artemisinin resistance. So this graph is from the published data that we have. And we can see that Cambodia, among all 3 countries, was the first one to detect with the artemisinin resistance. And by the end of the year 2015, as you can see here, all countries were detected with artemisinin resistance. And the number remained high until 2019. So this is just an example of the graph, the first graph that we produced together. Now, what if we want to change from the graph to map? Okay, what we can do is we're gonna go to our single map. So now we're gonna click on single maps. Okay, so now we're gonna see the display of the map like this. Is everyone having the same map? Okay. So if you wanna check and make sure, you can see on the side here that you have selected artemisinin resistance for the drug, and from the year 2000 to 2019 as your selection. Okay, so yeah, if you want to make sure, you can see that we get the sample from Cambodia, Laos, and Vietnam in the year 2000 to 2019. And yeah, this is the artemisinin resistant drug. So for the map, what you can do is you can adjust the size of the map. You can go to the setting here at the bottom left corner. And yeah, here you can choose to see by province or by district, but we're just gonna go with the province. And then you can whether shows the name on the map or the region. And you can also adjust the size of the map in case you want it to be bigger or smaller. In this case, I just want to reduce the size a little bit. So you can do between like 10 to 15. You can choose the size of the map. And then when you click display change, yeah, you can see that the size of the marker has been changed here. Okay. So we are having the same map here. So just quickly explain to you what this map means. Okay, so you can see that this map shows the level of resistance of artemisinin, right? And you can see that the number inside each marker indicated the proportion of sample with resistant markers ranging between 0 to 1. 1 means 100% of the sample carry resistant marker, while 0 mean no sample carry any resistant markers. As you can see here, I could take the example of here, 0.16 as Quantry province, Country province. So this means that 16% of the sample from this province were predicted to be artemisinin resistant. So that's how you can like briefly interpret this map. Okay. Yes. Yeah. We just, yeah, in the map, yeah, in the figure map. It's a small version. So, is your computer still work? Can we do another map? Okay, so now, what if we want to change from resistant map to the allele proportion map? So now it's just gonna change to a different type of map. So if your computer still works, let's try to do this together. Okay, so we're gonna click on edit here at the selected features. And we're gonna change from drug resistance to allele proportion. Okay, and now you wanna check which allele resistant to associated with artemisinin resistant. So we click on it again, edit, and then go to allele proportion. And then we know that PF-KELCH13 is associated with artemisinin resistance, so we're just gonna select that. Okay, I hope, yeah, this is your selection, allele proportion, and PF-KELCH13. Then click on display changes. Okay, so now you will see the map of allele proportion for PF-KELCH13 here. So same, you want to adjust the size of the marker. So you click on this here icon again. And for this time, you want to increase the size a little bit so you can see a different color in each pie chart. So you're increasing the size, 20 or 25, and then you click on display change. Okay, so now you will see this map here of allele proportion with different PF-KELCH13 alleles displayed here. Okay, I hope we get the same map here. Hopefully. So yeah, I just go through with you briefly about this map. So this map shows the proportion of all PF-13 allele, and we can see that the dominant allele during this period of time are Y type, which display in the blue color, and C580Y, which display in the red color. And we can see that the Y type is widely spread in Laos and Eastern Vietnam, and C580Y is widely spread in Cambodia and Western Vietnam. So that is allele proportion. So now you see the map with a different allele proportion in a different geographical location. Now, if you want to go back and see allele proportion of different years, what you can do is you can just change from the single map here into graph. So you just go back to graph. Okay, so now you can see the graph of different PF-13 allele displayed by years, starting from 2007 here until 2019. So I think we all can see the same graph, this one. So what can you tell about this graph is that in the early years, we can see that the Y type has been a dominated allele, while later on C580Y has been increasing over the year and become the dominant allele by the end of 2019. So this is what you can interpret it from the graph. So you can either display, if you want to see the geographical location, then you go to the map. And if you want to see like the distribution by year, you can just go to the graph. So yeah, you kind of like play with it a little bit. So now is the exciting time. So I would like to ask for those of you, that the computer still can work, to do a little exercise together. So I would like to ask for you to try and produce a chloroquine resistant map from East Africa in the year 2000 to 2015. So yeah, just make some simple sample selection and then try to produce this map together. So I'll just give you a few minutes to work on this. If you need help, just let us know and then we will assist you. When I try to produce that map, so you can see I chose the single map here on the options. And then for the country, I selected all the country in East Africa. So these are the list of the countries. And I chose the year 2000 to 2015. So this is my sample selections. And for the feature selection, I selected the drug resistant, which is chloroquine resistance. And this is how the map look like when I display the map. Okay. So we're gonna quickly move on to the compare map, which is the last thing that we're gonna be doing together. Okay, so now we had done the graph, and we also had done the single map. So now so now we're gonna do the compare map. So what you can do is you just click on the compare maps on the top there. Okay, I hope it's come out. Okay, so when you click on it, you can see there's like two maps side by side. For parameter A is the map on the left, and for parameter B is the map on the right, as you can see here. So we can adjust both of the maps. For example, for the first one, I want to change my sample selection. So I go to edit here. And I want to go back to my original selection. So I deselect all of the East African countries. That was all the exercise that we did before. Okay. Now I want to go back and select Cambodia, Laos, and Vietnam. Okay. But for this, I want to change my study time period. I want to compare map between two different study period. So the first one, I want to look at the map from the year 2000 to 2010. Okay, so this is the time period that I've chosen for the first map from the year 2000 to 2010. Okay, and then I click on display change. Okay, so that was my first map. But I also want to change the map features into PF-13. So I change to the allele proportion. And then PF-13. So that was my first map. Okay, let me just quickly go to the second map. Okay, so for the map B, we do the same thing. We go to edit and then deselected all the country that we have been selected before. And click on Cambodia, Laos, and Vietnam again. But this time, I want to change our study the time period into 2011 to 2019. So this is the time period that will be selected. And then we click on display changes. Okay, and same thing we're gonna do with the features. So we change from the drug resistant to the allele proportion, which is PF-13. And then we click on display change. Okay. Okay, so this you can see that the pie chart is pretty small, so we can adjust the size again. So you can go to the setting on the bottom of the page here, and then increasing the marker size. Yeah, I think it's just running again, the video. So for those of you who can't follow, can I just go to the end? Okay, so I'll just go to what we got at the end here. So after we have done our selection, so what I did is the first map we selected from the year 2000 to 2010, and the selected feature is the allele proportion PF-13. Same with the second map, the 3 country, but at this time the study period is different from 2011 to 2019. And the same feature of the map, which is PF-13. So when you compare this map, you can see that the wild type is dominant allele in Laos and border of Cambodia and Vietnam, while C580Y is widespread in Cambodia and eastern part of Vietnam. So this is, for example, you want to compare the same feature of map with a different period of time and see the distribution of the allele proportion for PF-13. So I know there's like a lot for these sessions that we go through together. So I just want to end with this. So if you need help, or if you want to go back and do more of our web application, and if you need help with this, I would like you can just contact Ethan here, who has been responsible for developing this app, web app interface, or you can also contact me and our GenRe-Mekong team, and we'll be willing to answer your questions. And just want to say thank you so much for your attention and participation. And yeah.
Thank you.
And leave a feedback too.